Okay, hi everyone. So we're going to continue on the discussion of the microorganism, in particular the prokaryotes. So we've looked at most interesting part, majority of the interesting part characterizing the bacteria last week. So this week, briefly, we'll go over another group of prokaryote, which is archaebacteria. The keyword for the archaebacteria is this: incredibly unfavorable habitats environment. Like we cannot even imagine that how can they live in such a high temperature habitat like 95 degrees Celsius. We cannot even most of the organisms actually cannot even live in a temperature something like above like 45 degrees Celsius, but they 75, 80 degrees Celsius, 95 degrees Celsius, even 100 degrees Celsius seems to be no problem for them, some of them. And also, extremely low temperature, like below sub-zero, minus 20, minus 30 degrees Celsius. Everything will be frozen, but some other archaebacteria, since they don't care, they can live happily there. And very salty environment. For example, this is the photograph of the Dead Sea, the famous Dead Sea near Israel. Nothing can live in because of high salinity. The salt concentration is too high, but some bacteria, for them, it is their happy, their original hometown habitat. So the question is, how can they manage surviving in such a, uh, normally speaking, non-livable environment? And continue on. This is a photograph of one of the hot spring in the Yellowstone National Park. The temperature around here would be extremely hot. So almost near boiling, but some bacteria can grow happily there. And this pinkish layer on the ground are actually layers of bacteria. And frost ground, this is a sub-zero, like maybe something like minus 15 degree Celsius. Everything is frozen, like I said. But some bacteria can grow there very actively. And this is some bacterial population can even grow inside of our stomach. The environment of our internal stomach condition is something like a pH uh, about pH 2.0 below, which is a strong acidic condition. Like everything will melt. That's why we can easily digest all our food product inside our stomach, but some bacteria can grow, no problem for acidity. And the opposite can be true for some other group of bacteria. So to give you several examples of how such an incredibly harsh environment uh, seems to be no problem for some archaebacteria. So characteristically speaking, generally speaking, we can put some keyword for archaebacteria: extremely harsh environment. And so more important question is this then. Yeah, so it is really amazing to observe some incredible creatures can live under such an extremely unfavorable environmental condition. But more importantly, this is like, how can they do that? Somehow, some their structure, of course, some particular structure of them, notably like they're outside the cell wall. Remind you, bacteria have all cell walls, yeah? So maybe chemical composition of the cell wall might be slightly something different so that this may be able to provide some extra protection under such difficult conditions. Or what about cell membrane inside of the cell wall? Is this also same in terms of chemically, like the cell membrane structure that we know of, like a phospholipid bilayer, or is there something different? Might be some legitimate question that we want to put on after knowing their incredible ability of managing such difficult environment, but also this the ability to, like, there must be some specific enzymes. After all, enzymes are one. That's why I jokingly compare this enzyme as a magicians. Enzyme can do everything, for example. On the other hand, without enzyme, we cannot do anything. So there must be some special enzyme live inside of this very weird organisms. So that could be another kind of a perspective that we might want to look at. And that is actually the value, the most interesting feature and the value of this archaebacteria for us to study. Of course, a pure academic, scientific curiosity, yes, is probably the most, in textbook definition, should be the most important things. But in reality, all these scientific interests are mainly, a lot of times actually, driven by some other applicative benefit. If there is something that we can get as in return, then we really jump in harder. And that is the case for this archaebacteria. This enzyme, incredible, unusual ability of this enzyme, metabolize highly unusual substances to extract energy and etc. This is only one example. So, very unusual unusual ability that several enzymes can showcase is another very interesting point to which many scientists are actually trying to figure out. So one very famous example of such enzyme, very special ability of the enzyme related with this archaebacteria is this. Right now, almost everyone knows PCR, because like PCR stands for polymerase chain reaction. That's what it stands for. This PCR is nothing but the technique by which certain defined area of DNA can be amplified, like made in many copies. tremendously many copies in a short period of time is the technique, the essence of this technique. But using this PCR technique, we can do so many different things. So that technique has benefited so much on many, many areas, including medical science and forensic science, trying to identify some, like some saliva and blood stain with such a small, tiny amount of a sample. In the past, it has been impossible whatsoever to identify who's this. Someone left some small, tiny blood stain, then how can we identify this blood sample belongs to whom? But using this technique, because from scratch of small, tiny sample, we can amplify the DNA in a huge amount, and it is no sweat that, oh, this DNA sequence belongs to that guy or that person or anything. So forensic science is the one that we can, in everyday our lives, actually, we can hear so many incidences that have been solved, and that belongs to the many cases of the forensic science. But also medical diagnosis. When we try to identify some specific cause for the specific diseases, also the PCR technology can also provide us so much convenience and efficiencies. And one recent example, corona testing, is based on this PCR. There's also another such an example with very small, tiny traces of the virus, like virus genetic material. It can be applied to both DNA and RNA. Okay? And we can amplify the amount in so great amount so that, oh, this sample. So without it, probably we would have really faces such a technical difficulty to identify, even if someone was contaminated or infected or whatever with such very small amount of viruses. And in that case, it would have been impossible to detect or make a correct conclusion whether this person was infected or not. But using this PCR tests, highly accurately, with very small amount of sample, we can do it. And those are not even the every story about this PCR application. There are so many different areas this PCR can be used. So no wonder, again, I thought this computer has solved this problem, and it start to scare me again. So anyway, so this PCR, but the beginning of this PCR technology was the identification of one particular enzyme from one of the archaebacteria. The enzyme responsible for replicating DNA, we call a DNA polymerase. Everybody has DNA polymerase, including bacteria, human, plants, and whatever. Everybody, every living organism must have their own version of DNA polymerase. And that's how we actually look at one of the antibiotics is actually targeting on this DNA polymerase of prokaryotes. 2 weeks ago, we have looked at it. But this DNA polymerase is extremely interesting from that archaebacteria, in that this DNA polymerase doesn't even die at 100 degrees Celsius. Most of the enzyme will be destroyed at something like 42 degrees or 45 degrees Celsius. But this particular DNA polymerase extracted from this archaebacteria, actually the optimum temperature for its working is something like 80 degrees Celsius. And 100 degree boiling temperature, it doesn't die. which means it is not destroyed at all. So the person who discovered this enzyme from this archaebacteria and then decided to use this technology must have earned a lot of money. Yes, that is the case. He became a billionaire. Yes. Literally billionaire. He couldn't even count his money. And what's more, usually as a scientist, if you uh, get a benefit from huge profit, monetary reward, then your fame as a honor, as a Nobel laureate, usually you better forget it. Okay? You already had your own share of your rewards in terms of money. So why do you want to have another honor in terms of a Nobel Prize winning things? But this guy had both of them. After about 10 years of becoming a billionaire, even, people cannot deny the impact that PCR technology has brought to us. So there is no way for us to deny and then, oh, we have to give this year's Nobel Prize to that guy. Although we really, really hate to give this to one of us, such a billionaire, a drug addict billionaire, actually, it turned out that. But anyway, so that was the case. In other words, this archaebacteria, so there must be, it should not be the only example of the benefits of this researching the archaebacteria, obviously. So there must be a ton of other valuable and interesting enzymes that which we did not even yet discovered. And that's already still nowadays, still again, many scientists are still looking for some such possibility of another gold mining of this, some exotic enzymes from archaebacteria. Is probably the story that we can leave. With that, we can leave the archaebacteria. So, oh, another thing is, yes, I almost forgot. Although it's not really, it is simply a trivial thing. So if you want to forget it, yes, you can forget it. But this is a typical structure of the cell membrane. That's why we call this as a lipid, the phospholipid bilayer. Top layer, bottom layer, so two layers, that's all phospholipid bilayer. And this is fatty acid chain and phosphate groups and some hydrophilic things, a typical chemical structure, isn't it? But archaeolipids, some portion of archaeolipid is even very interesting in that this two, the fatty acid tails are fused together. So for this, we cannot call this as a phospholipid bilayer anymore, right? This is a monolayer, fused. Or the other thing is actually there are some detailed chemical compositions are also different from the typical, conventional, the lipid bilayer things. And this is something to do with, let's go. This is something to do with the heat resistance of extreme heat resistance of archaebacteria. In a computer simulation, we can see easily that some heat, in a minute, some heat was applied to conventional bacterial cell membrane as well as our membrane too. These whole membranes are like miserably destroyed. But as you can see, this high heat doesn't even do anything about, at least for the moment. So this shows that this type of structure of cell membrane of archaebacteria can confer the really stronger resistance against the heat. So maybe, at least maybe some bacteria that can live in a hot spring or a volcanic area. Maybe, yeah. At least part of the reason why they can do that is because of this unusual cell membrane structure. And this type of differences can be extrapolated or expanded. So there must be some other differences in terms of their structure, cell or cell wall. Yeah. Actually, I didn't bother to discuss, but their cell wall structure is also different. Yeah. The archibacterial cell walls are also different, but yeah, big deal. At least I'd like to show these cell membrane structures differences to showcase, exemplify that, yeah, actually there are some structural differences that may be able to explain their unusual behavior for the archibacteria. Not to mention, of course, there are several enzymes, many enzymes that also enable their unusual ability for survival is probably okay. So, yeah, that should be. Now the final part of this chapter is about, as I advertised in the beginning of this chapter, now we are going to briefly look at the techniques that enabled us to be able to enjoy such a huge and the rich knowledge of internal structure of the cell, which is the microscope. Yeah. Without it, yeah, as I said earlier, it would have been literally impossible at all. So in visualizing cell, the microscope, and here first, before we go over several different the slides of the lecture that I have prepared, let's once again try to summarize some keywords here. First, light microscope. The light microscope is the general microscope that you probably almost every one of you have an experience to see once in your lifetime. Until now, probably you have looked at it somehow in your high school or somewhere else, probably because it is small, relatively small, tiny, relatively speaking, and it is cheap. So this light microscope is one kind. The second kind, which is different from this light microscope, is we call EM, electron microscope. So the difference between these two groups of microscope in terms of their like principle is what kind of a light source they use. In order to visualize, we must have some lighting source. In the case of a light microscope, yeah, obviously this light source is the visible light. Along with so many different wavelengths of light, we have only very narrow limits of visibility, and that narrow limit of light wavelengths is the one that we can utilize in examining any specimen under the light microscope. So we need to have some light source for the visible light source for light microscope. Okay, but the electron microscope, they do not use the light as their lighting source. They use the electron beam. Okay. So it is kind of a digital light source, so to speak. And also, accordingly, this light microscope, we have this lens. Okay. The optical lens, which is made of basically the glass. Okay. And depending on the different depths of this lens, this visible light can be reflected with varying degree. That's how we can magnify the image. The thicker this lens, the higher the magnification can be. So that's the principle. But in the case of an electron microscope, since we are using the electron beam, so this lens can also be some kind of a digital lens. So the magnification can also be extremely higher than that of the light microscope. So these two. And regardless of this, whether it is a light microscope or electron microscope, for successful examination of any sample under the light microscope, there are two different parameters actually are involved in successful examination of any image under light microscope or any microscope. The first thing is magnification, of course. How large can you magnify any specimen? So that's why we need a microscope if you want to look at very tiny bacteria, which in average, the average diameter of any prokaryotic cell is 1 micrometer. Can you imagine how small the 1 micrometer? 1 micrometer. You cannot even see that small spot. So that's why we need to get any help from this magnifying power of microscope. So this magnification. However, another independent parameter that is also important is the resolution. As you go higher in the magnifying the area, then actually you lose this resolution, because as you can easily understand in these days, the resolution, because of this all different TV monitor and computer screen that advertising the superior resolution. So resolution means how like a small any single dot in a screen that can fill is the resolution. Under the microscope, the resolution is like let's say there are two lines. In reality, if you increase the magnification under the light microscope, at some point, these two lines, the thickness of these two lines become larger and larger. So at some point, these two lines like overlap. So you cannot even distinguish these two lines separately. Then that's the point when you lose the resolution. So that's the kind of the definition of a microscopic resolution. So in other words, because of this resolution limit in reality, because of this resolution limit, there is so much that you can go like achieve the higher magnification. There is only so much you can do, and beyond that, you will lose the resolution. And this is particularly true for the light microscope. And so this is the diagram that tries to explain what's the problem of this resolution. If you increase this thing now, these two lines, the distance between two lines will become blurry under the microscope. So now you don't have any meaningful any image under the microscope. That's the resolution. So magnification is good, of course, but another thing that you need to actually consider is this: can you keep the fine resolution with such a high magnification. And this formula, which is, all right, let me just briefly explain. This D indicates the resolution, and the smaller the D value, that means it is a higher resolution. So you try, what you want to do is you try to achieve the smallest ever possible D value, which is controlled by these two parameters. One is the lambda, which is wavelengths. Okay. In the case of a light microscope, this is simply, there is no way you can control because you have to use your visible light source. The visible light source wavelength is fixed. Are you following me? Okay, whatever. Yeah. So in other words, yeah, this one, you cannot do anything. The only thing that you can try to ever control is this. The NA, standing for numerical aperture, okay, which is also kind of constant. Nothing you can do about it because in the air, the numerical aperture value is almost like usually near 1, whereas through the oil media, it has a slightly higher value. In other words, actually, it's slightly better if you try to examine your sample under oil. Okay, this is not for everyone, but especially for only for some of the science major in the laboratory class. Then you have 20, 40, and 100x magnifying power on your microscope. Usually these two, 20x and 40x magnification, you do not have to do anything else. Simply rotate, okay, revolve your objective lens, that's it. But probably, at least some of you may have experienced that when you try to move your the power of magnification into 100x, then everything became blurry. Nothing can. And if you had your assistant, like a laboratory assistant, lab TA, teaching assistant, then probably she or he would like to help you, like by advising that, hey, for 100x magnification, you have to put oil. What do I mean by that? Oil on your slide under the microscope, cover slip, there is a cover slip, and your specimen sample is on top of the slide. But for this 100x magnification, you have to have 1 drop of oil placed in between your cover slip and the slide. And afterwards, you put your slide on the 100x the microscope. And now become everything is much clearer. That is the reason why. In the oil, okay, oil, your resolution has an improved resolution. That's why on the higher magnification, you better use some different numerical aperture is the reason. So as a science major, if you've been wondering, why do I have to use oil in the higher mag? That is the reason that you can understand. For non-science major, forget it. Why do you have to understand this thing? I'm not going to ask you this in the exam. This is only just in case for those science major that you've been wondering. But okay, before I jump into the electron microscope, let me introduce a special version of light microscope, which is fluorescence microscope. This fluorescence microscope is a special type of light microscope, of course. Instead of some differences in here is instead of using visible light for light source, they use the invisible area of a laser light or some non-visible area wavelengths of the light also can be applied over here. Instead of trying to explain some principle of fluorescence, let me just simply put that the nature of fluorescence is not every molecule can become fluorescence. Only some special molecules can be fluorescent. Those fluorescent molecules can absorb one particular wavelengths of light, absorb, and then emit that energy into different wavelengths of light. That is something very special ability for such a special molecule. And then we recognize them as a fluorescent. And this fluorescence microscope use such fluorescence as a like indicator of the sample. So depending on the type of molecule and depending on different wavelengths of this light that this molecule absorb, that the kind of color of light that we can detect coming out of this fluorescence molecule can vary. Sometimes it's red, sometimes green, some other times blue, and some other times yellow, and all different array of rainbow color fluorescence light can we detect. That's the beauty, literally beauty of fluorescence microscope. So in other words, if you happen to have any one particular molecule or structure of interest, then you can apply, if we can apply this fluorescence technique to that target, then we can follow only that molecule on the light light microscope. So, like this. This is another such example of the different color of fluorescence. Like the nucleus can be labeled as a blue fluorescence, whereas some other cytoplasm, some other structures can be stained, like fluorescently stained with green, etc. So in other words, once again, try to explain in very simpler term the fluorescence. If you have any particular target, then if you can attach some specific fluorescence tag on this particular target, and this will glow under the microscope, which has to be a fluorescence microscope. On the microscope, this particular target only will glow particular colors of like this green or blue or red is the principle of a fluorescence microscope. Okay. So you can forget about everything else, but if you can only follow that your target of interest, which has been attached. We technically attaching the fluorescence dye to that particular target, we call it as a label. Labeling. We label your target. Let's say this is your target. And there is a very technical way that you can attach or label this your target only with this fluorescence tag. Okay. And thanks to this fluorescence tag, which was labeled to your target, then now everywhere, whenever this target go in the cell, under the microscope, you can follow this particular molecule only, following this fluorescence color. Is the principle of a fluorescence microscope. Okay. Now, among this fluorescence tag, there has been breakthrough some time ago. This is the model, structural model of a particular protein whose name happens to be GFP, standing for green fluorescence protein, GFP. And this protein, we do not have this protein. Okay? Only this protein has been found, this jellyfish. So, very similar to that archaebacteria happen to carry that weird polymerase. This weird jellyfish glow in green thanks to this GFP that this jellyfish carry. And we once again, what can you expect? We once again steal the gene of this GFP from this jellyfish without any permission or thanking. And we decided to use this in labeling. This is a protein, and this is what a convenience. This is a protein, not a chemical. Unlike most conventional fluorescence dye, which are basically chemical, there are some kind of inconvenience because of the nature of this chemical. But this is proteins. So this is tremendous advantage comes with this protein-based fluorescence tag, which is we can fuse this protein with our own interest, molecular interest, like I didn't even include this. Okay. What a disrespect for you. I did not even. Sorry. I'll have to explain my poor wording. Let's say this is YFG, your favorite gene. Okay. So whatever it is, it could be an insulin or any kind of cancer-causing gene, whatever. So this will be make a protein. So let's say this is a protein. Then you can combine the green fluorescence in the tail portion, so. This part will be GFP. So all together right now, you create some chimera. So now the whole fused protein, fusion protein containing green fluorescence protein part. But this protein, after this fusion, this protein still will be functional, will be normal in most of the cases. Now, if you put this gene back into the cell, this is gene, the chimera gene, you put back in, then now, because of this green fluorescent protein part, this protein will glow in the cell. Before, this protein was invisible, colorless. Now, because of this kind of some engineering, editing, this your target protein, whatever it is, your favorite gene protein, will glow this green color. So you can follow everywhere, whenever it goes. A lot of times, actually, the first step that if you dedicate your life in researching any particular gene or protein, the first step is where does it go in the cell? Does it go to the cytoplasm, or does it go to Golgi, or does it go to the nucleus or cell membrane kind of stuff? That's one certain first lines of questions that can be easily answered without any artifact. Is the beauty of this, bless you, green fluorescent proteins, the benefit. So, so many different variety of this GFP has been developed. So nowadays, this GFP is not green anymore. There are several different color versions have been developed because of the tremendous financial motives that are there. And obviously, guess what? The three scientists who developed for the first time this GFP in this technology also won Nobel Prize recently. So, uh, so anyway, once again, what is this is once again the keyword for the fluorescence microscope, live imaging. Under the light microscope, observing some cellular structure is to first destruction, all different part of the structures, and you cannot pick only one such molecule of your own interest that to target is one drawback. Another thing is usually in order to examine with high resolution under light microscope, you have to kill the sample because you have to stain them. Without any staining, coloring, there's no way you can observe this. Remember, gram positive, gram negative staining? Yeah, you have to stain them. But once the minute you stain the sample, the cells die. And so you are simply beating a dead horse. How can you expect any live activity? You are interested in actual dynamics of a cell function using the dead cell. But this is a different story. Using this fluorescence microscope, especially the GFP, because you can imagine and you can trace your particular target in the living cell once you put this gene back into the cell under the microscopes. That's why we can dare to declare that the advantage of this fluorescence microscope is this live imaging, which is extremely important feature. Anything else cannot provide such advantage of visualizing any live imaging, which is very important, significant, as you can easily imagine. So that's it. And also technologically speaking, the resolution, today's fluorescence microscopes technique has such a tremendous progress in that even the resolution, you can see the different resolution level of the top and bottom panel. And this top panel's resolution is the today's resolution level. And even the above one was the old-fashioned fluorescence imaging resolutions. You can forget about that. And in these days, you can enjoy such a fine detailed image of a fluorescence microscope. And so even you can individually distinguish some of the very small, tiny structure. This has the size of this particular structure can be even comparable to a ribosome. So if you can think, you can distinguish individual ribosome in a cell, wow. But now fluorescence microscope can do that. So almost you can as a morph into another dimension of this area of technology. Look at how beautiful is this such fluorescence microscope. Now the electron microscope using electron beam. Yeah, sure. Although I did not include the advantage of electron microscope in here, this same formula can also be applied to the resolution of electron microscope in that this lambda can no longer be a light wavelength, but this is electron, so it can be extremely small, okay? this light source, electrons, and also, so you can forget about this numerical aperture over here. No way. Depending on what kind of voltage you use, the lambda value of electron microscope can be something like 0.000000 something, which can be translated into resolution. So electron microscope can have a non-comparable resolution. Extremely fine high resolution is what this can say about it. And also magnifying power is also tremendous. So that's why electron microscope usually can present this type of fine detail: chromosome, chromatin actually, and nucleus, nuclear membrane, and endoplasmic reticulum. Wow, this type of electron microscope can do this. Light microscope, forget it. You cannot. Maybe today's high-tech fluorescence microscope may be able to approach this high resolution, but in the case of a fluorescence microscope, you have to apply different fluorescence dye in every different structure. That could be another disadvantage in the case of this fluorescence microscope. Okay. Look, this is a virus try to infect the poor bacteria. This virus is called the bacteriophage, which infect only the bacteria. And and this is mouse part of a fly. You didn't know this really detailed and nice structure of a fly that and where is okay? Is there any? And this is another pollens and red blood cell and white blood cell on the cut part of your blood vessel. One disadvantage traditionally of electron microscope has been that it can only generate the black and white image because of some technique that involved in. And another thing is more disadvantage, actually, more serious disadvantage of electron microscope, which is, once again, all this structure that needs to be examined under electron microscope must be fixed, which means you cannot examine any living cell. So they are all dead cells. And also, not only they are dead, but due to this structural, the specimen sample preparation technique, these artifacts. So the structure that you see under the electron microscopes are actually you begin to wonder whether these structures are really real or some kind of artificial image that never exist in nature, but due to this sample handling somehow distorted structures. A lot of times, actually, that problem occurred involved with this electron microscope sample. But even with that, as you can see, the fine detailed structure that observable through this electron microscope, you cannot even deny such advantage in that. Now, the electron microscope, which is relatively very old-fashioned, is not the focus of today's lecture's discussion, actually. Today's lecture's discussion point highlight is this. Another newer version of this electron microscope called the cryo-EM. Using the conventional electron microscope, simply some scientists thought about that. Can we use this electron microscope in different way? instead of examining the whole cell structure as we used to do it, but can we use this electron microscope in something different like protein structure? Protein structure is, I try to emphasize the importance of this protein structure whenever possible, because knowing exactly how any particular protein looks like is extremely, extremely important for understanding function, and also trying to develop some drugs targeting this particular part of structure too. So traditionally, several different ways of solving the problem of knowing protein structure. But such several technology all have their own advantages and also disadvantages. So that has been limited. The advancement of this understanding the function of a particular protein. If you want to know the function of a gene, for example, we have actually 23,000 different genes in our human genome. It's not that many, much fewer than we have originally expected, but still 23,000 genes, each of which that can make protein is a lot. But a lot of such 23,000 genes, they are all protein coding gene, protein making gene. We do not know their functions. What do they do? It's strange, isn't it? At this area, 21st century, we still do not know what our particular gene's function is, and that is the reality. So in order to understand the function of a particular gene, it's extremely important to understand the structure of this protein. But there is no way we can know the structure. It's technically very difficult to figure out the structure. So if someone can actually present another way of manipulating the structure of a protein, that would be really nice. And some scientists tried using. So cryo means extremely low temperature. Cryo, low temperature. So under this technique, try to prepare the protein samples in extremely low temperature, something like minus 300 degrees, which is extremely low temperature. And then spread those samples in some area and try to examine under the electron microscope. To explain simply, this is in principle, this is what it is. Okay. So you prepare same copies of protein in many copies in a small chamber. And each protein, okay, they were quickly frozen and then they are fragmented. Proteins fragmented. And in this area, the area of these proteins, or every different possible area of these fragmented proteins were exposed. And then they are laid out. And you are trying to visualize this different area, part of this particular protein under the electron microscope. So whole different part of the protein, panorama like this protein profile has been generated. Now it's computer's job to figure out and assemble this different fragment part into the whole shape of a protein. Is basically the principle of this cryo-EM. So whatever it is, it's been tremendous success to the point of these three gentlemen were also able to get Nobel Prize. So everybody gets Nobel Prize. Why not me? Jesus. The protein structures resolution has been tremendous progress. So before like 2013, the resolution of protein under cryo-EM has been like this. So it is not so much of use. But in these days, the resolution of a protein structure reached to the even individual amino acid level. So every fine details of a protein structure can be resolved, figured out using this cryo-EM. And it is really tremendous breakthrough. So this is a very famous case of like when we try to, not we, but when scientists try to develop the antibody, effective antibody against the coronavirus, then you have to first pick a target, particular shape, target area. This is the shape of coronavirus. And this particular target, which we call the spike protein, has been selected for like antibody production. So antibody will specifically recognize this part of the coronavirus and then we'll try to grab. As that's how we develop. I keep saying we, not me, but that's how they developed these antibodies, right? So then, in order to do that, you have to be able to figure out the structure, shape of this coronavirus. And cryo-EM, without this cryo-EM, the more conventional way of figuring out this structure, probably it would have taken several years. And even with that, the chance of success is not really great. But this cryo-EM, you can do this thing within a matter of like month. And that's what they did. So quickly figuring out the particular structure to be used as a target. See, this fine details target has been resolved. And now you can now develop this particular antibody, which antibody will be able to bind, interact most effectively. Also, you can determine using this under this cryo-EM figured out structure on the computer simulation. That's how you selected the most ideal antibodies, and it's been used with the help of another vaccine technology, which is DNA or RNA vaccine too. So I'm not really sure I have mentioned before or not, but the artificial intelligence also has been another area of this the protein structure provider. This artificial intelligence in these days, especially the Google's Deep Minds company generate this alpha. There is particular AI called AlphaFold. You can easily, any general public can have an free access to this AlphaFold AI. And this is the particular AI that predicts protein structure. And this computer-generated prediction, the difference of this different feature of this AlphaFold AI is that the accuracy. Before, the prediction has been nearly almost impossible because of so many different possibilities, but somehow this AI solved such barrier, technical barrier. So right now, it's been considered and it's quite accurate, extremely, it's very accurate. And with that, AlphaFold AI prediction of a protein structure together with this cryo-EM technology that can confirm this predicted structure this area of protein structure can really progress very quickly, so that many problems that viewed as impossible to solve can be handled, taken care of easily in very near future, is the kind of future that we are facing now. Especially, like, you can develop so many different drugs. Even today's newspaper, I read that some group of scientists developed new antibiotics, okay, targeting on some superbacteria. No existing currently available antibiotics can work for this particular superbacteria, but AI predicted some particular structure that can particularly target on this particular superbacteria, so that design and then produce this particular drug is another such example of this research area. Okay? Uh, so, yeah. Thank you. And we can take 25 minutes and then continue on the new chapter in the second session.